Hello, everyone. This is Sabrina Paganoni, and I'm here from the HEALEY Center at MGH, and I'm giving everyone a few seconds to join from the meeting room, from the waiting room. And as always, we are providing you with this weekly update, and we're always happy to have guest speakers. So tonight, I'm really excited that I'm not here by myself, only me and Catherine, but or other usual panelists, but really we have one of our site PIs, Dr. Nicholas Maragakis, who's also a scientist and expert on a number of ALS topics. I'll let him introduce himself, but essentially we're so happy to have him as guest speaker to tell us more about his center and also about the important topic of biomarkers. Dr. Maragakis?
Thanks, Sabrina, and thanks for asking me to participate. It's been it's been a great opportunity to be part of the HEALEY platform study, both as an investigator and to also help move some of these drugs to participants. So it's really been a pleasure, and I'm happy to talk a little bit today. I'll tell you just a little bit about our site, but then we're going to talk a little bit about biomarkers and maybe update you on some exciting new work. So I'm at Johns Hopkins. I'm a professor of neurology, and I have a basic science laboratory interested in ALS, and we use induced pluripotent stem cells to study ALS. And then I also run clinical trials and see patients with ALS as well. Next slide. So we really have a wonderful team at Hopkins. I would be remiss if I didn't comment on this team. We have two large clinics, one sponsored by the ALS Association and one sponsored by the Muscular Dystrophy Association. And so that allows us to see really hundreds of patient visits per year with just really outstanding professionals in a multidisciplinary care setting. Next slide. As part of that, we have again, a really outstanding team that's been really key in taking care of the HEALEY platform trial at our site, as well as other clinical trials really that are afforded by a number of company-sponsored trials as well. And so this includes nurse practitioners, research coordinators, a number of physicians, all as part of our ALS clinical trials, which we're really proud of and launched as a new specific endeavor a couple of years ago. So this has been a terrific team, and we're really happy to be part of this effort. Next slide. I want to comment a little bit about biomarkers today. And as many of you know, the HEALEY platform trial is really a unique opportunity to advance science. Not only does it allow the ALS community as a whole to screen or study drugs more effectively and more efficiently as part of a larger community, but it also allows us to really advance science. And so we often talk about bench to bedside, and I would argue much of what we're doing as part of the HEALEY platform trial is bedside to bench, and that is that what we learn from participants in this trial will also help us really design better drugs and better studies. And so I think that's really a back and forth effort. So as part of this, DNA, we're collecting DNA to try to understand if certain patients with certain genetic forms of ALS, or if there are certain markers that might they benefit better than other patients who have a different genetic makeup. You may have heard about this nerve protein called neurofilament, and it's really exciting because it's probably a marker for nerve cell death in ALS and other diseases as well. And so the HEALEY platform trial is collecting neurofilament in all of their studies. And so can this inform us as what we call a biomarker, that is a marker of disease progression, and hopefully a marker of how drugs might be working. And this particular neurofilament is really part of a larger topic, and that is biomarkers. And a biomarker is simply something in the blood, in the urine, in the cerebrospinal fluid that can inform us, again, about how disease might be progressing, about who might respond to a particular therapy, And if a particular therapy is working, does that biomarker change? And that's not unique to ALS. In fact, there are very good biomarkers for other neurological diseases as well that can help us run clinical trials more effectively and get to answers more effectively. There are other things in addition to blood and spinal fluid and so forth, and those include things like speech analyses, or what we call digital biomarkers. And this really has taken off since the COVID-19 pandemic. And that is, as many of you may know, can we understand how ALS progresses, or how a drug might work more effectively by doing specific analyses like speech, or like strength, or like breathing function at home? And can we do that more efficiently and potentially more often? And so these are all different kinds of ways that what we hope to learn through the platform trial that can inform us, we think, about how to design better drugs for ALS. Next slide. So I want to speak briefly on one particular topic because it's kind of a hot topic currently. And I'm going to do a little bit of science, but I'm going to keep this, hopefully that will offer a broad perspective. There's a particular protein called TDP-43, and it is an important component of ALS pathogenesis and probably other neurological diseases as well. You may have heard of the movie Guardians of the Galaxy, and one of my colleagues has called TDP-43 a guardian of the transcriptome, and that is the TDP seems to oversee how proteins get made. And normally that protein should sit in the nucleus, as you can appreciate here on the left. So it should stay in the nucleus normally. But for reasons we're trying to understand in ALS, that protein leaves the nucleus. And so if it leaves the nucleus, it can no longer perform its function effectively. And in fact, if we look at brain and spinal cord of ALS patients, when that protein aggregates in the cytoplasm, it forms these bundles of protein that are abnormal. We can see that in the middle section here, that this protein, where you see the arrows, gets mislocalized. Now, if you knock down this particular protein, you can see here on the far right, it's called a volcano plot. If you knock down this protein, you get a whole bunch of changes in the transcriptome where it affects the cell in a variety of different ways. And so when you knock down this protein, what other proteins are affected? And we're starting to we as a community are starting to try to understand what other proteins or what other RNAs are affected. And can that lead us to better diagnostics? And TDP, I'll tell you, is particularly important because it seems to be common among almost all forms of ALS, sporadic ALS, most forms of genetic ALS. So we think it's a key protein in how all of ALS may work. Next slide. So with this in mind, TDP-43 loss of function seems to be a key mediator of ALS, both SALS, sporadic ALS, but oftentimes genetic forms of ALS, like what we call C9 ALS. And there are two important papers that are coming. One is highlighted here, and I'll tell you why that's important. This is what we call a preprint, that is, it hasn't been published yet. This is by a group at Johns Hopkins. And what it suggests is that as a guardian of the transcriptome, when TDP-43 doesn't work right, proteins aren't made properly. And you can see these are called cryptic exons. And you see this term cryptic here in red. And it means that the protein isn't formed correctly. And this group found one particular protein called HDGFL2. It's not, the acronym isn't important. But what is important is, as you can see in part A, is that in control patients, this protein is fairly low. But in sporadic ALS, this abnormal protein is higher. In patients with the C9 gene who don't have ALS, it's even higher. And in patients who already have or are symptomatic and carry the C9 mutation, it's even higher than that, suggesting that this abnormal protein that's formed continues to, it goes up in the presence of ALS. Now, what's really exciting is Dr. Wong and his colleagues actually used neurofilament and made a ratio, and that is the neurofilament ratio to this abnormal protein ratio. And so I told you earlier, neurofilament's important. In pre-symptomatic C9, if they use that ratio, it's very, very low in patients without ALS, but who carry a gene for ALS. But once they become symptomatic, you can see that that ratio changes really significantly, becomes much higher. And so that's particularly relevant in the context of ALS, we think, both pre-symptomatically as well as symptomatically. Next slide. So this isn't just a one-off. It's actually been nicely also in a preprint article that has yet to be published by colleagues at the NIH. They took a slightly different technique and identified different peptides, but also reproduced Dr. Wong's work from Johns Hopkins. And they used a different technique, something called mass spec, to identify some of these cryptic peptides. And so they found another whole group of these peptides, and they found some specific biology, but more importantly for us as a community, they found it again in cerebrospinal fluid of patients with ALS. So now this is two groups using two different techniques to identify these cryptic peptides in cerebrospinal fluid for ALS patients. So we believe that this could be, because TDP-43 and these cryptic peptides are probably present in sporadic ALS, this could be some of the kind of next greatest discovery, if you will. Now, what I'll say is lots more work to be done, and both of those groups would admit that. One of the shortcomings of these papers is that there's dealing with relatively small numbers of samples. So next slide. So what do we need to be doing? What's the next step? So there are multiple TDP-43 readouts coming. I showed you today these cryptic peptides that can be detected by a very standard method that's called ELISA, that's used across the community to look at abnormal proteins or proteins in general. But as we dig down, we'll probably be able to look at what we call RNA analytics. That is, dig even deeper into what changes occur with this TDP-43. Well, what do we need to be doing as a community? These are two particular, what we call cryptic peptides in two particular studies, but there are lots more that are likely to come. There's lots more to do. These results need to be validated and need to be fortified with more and more samples. And that's particularly important because it's, as part of the HEALEY platform study, it's going to be really important. We ask participants to, if they can, to get lumbar punctures to take CSF. And this is exactly a reason why this is so important, to get CSF so we can increase the number of samples we have, so we can look across time parameters, that is, to see how these particular cryptic peptides change over time, and importantly, what effect drugs might have on parameters like these. So we're already used, our colleagues used banked CSF, that is, that was already in the freezer, both from this study and others. But can we expand that sample? And that's going to be really important. We also want to correlate it with other disease parameters. Again, does it change with disease progression? Do some patients or some participants who have, let's say, bulbar ALS, do they have more of these cryptic peptides than patients with limb onset ALS? Does age affect it? Does gender affect it? All these are key components. And again, importantly, do these cryptic peptides change in response to the therapeutics that the HEALEY platform trial is bringing to the fore? And finally, can we correlate these with existing biomarkers? It's probably the case that none of these biomarkers exist in a vacuum. Can we mix and match them to get a better panel of biomarkers? And these are just really two examples or three examples of biomarkers that are being used in this context. But we and others are also looking at other potential biomarkers. And so that's really a mission of the ALS community, both as part of the HEALEY trial, nationally and internationally, to try to expand our understanding of biomarker development. So I think that's my last slide.
This was such a great overview. And just to kind of wrap this up before we go to questions again, as Dr. Maragakis was saying, in addition to testing several drugs, One of the goals of the platform trial is really to advance science so that we can understand ALS better. And if we can develop better biomarkers as part of this research, then we're all going to be better positioned, essentially, to adapt the trial, design new trials, better test drugs. So I would like to thank all the participants, past, present, and future, who are really contributing a lot of their time and also a lot of procedures and kind of, donating blood and spinal fluid so that we can really continue to progress and validate these discoveries. If we can go to the next slide. Just wanted to mention that we are excited to start enrolling for regimens F and then G very soon. Right now, we have 11 sites that have been activated for regimen F, so we're starting to screen participants, and again, regimen G will start very soon as well. So we will have 2 drugs being tested in the platform trial very soon. At the same time, next slide. And for regimen F, which is open right now, we do have a number of resources on our website. The QR code will bring you there. And as always, please feel free to contact Catherine for more information. But essentially, a key component of this regimen F, as well as upcoming regimens in general, is to get spinal fluid by doing 2 lumbar punctures so that we can measure not only this, but several other biomarkers, in addition to the ones that were presented today. That can really tell us a lot about the drugs and also can help us develop better tools for ALS in general. Next slide. And as always, again, feel free to share information about this webinar. We're going to have another biomarker discussion on March 23rd. Again, we really want to make sure that we are available and we get all the great scientists doing work on this. Today, very pleased to have had Dr. Maragakis and his colleague also from Johns Hopkins, Dr. Rothstein, will be on this webinar next. So again, we can have a forum where everyone can ask questions about the latest news in biomarker science. And I'm so happy that also we will have an extended webinar on March 27th, which is not a Thursday, actually, that's an additional webinar. So we'll definitely post that on the web. Please feel free to invite everyone who would like to learn more about regimen F, again, the one that's enrolling right now, and then we'll do the same for regimen G over the next few weeks. And with that, I think we can take down the slides. And Dr. Maragakis, I'm pleased to see that a few members, a few people who are attending are actually following your research. And there's a couple of questions about your research in particular. So one question is, I believe you studied a drug called tonaberstat. Can you tell us more? And are you going to do a clinical trial about it?
Thanks for the question. Just briefly, we've been very keen on trying to understand how a cell type called astrocytes can affect disease progression actually after onset. And we identified a protein called connexin 43. And turns out there's a drug that has been used in migraine previously called tonaberstat, was used years ago. And we were able to show that, in fact, using iPS cells, iPS cell-derived motor neurons and astrocytes, that drug seemed to be effective in slowing cell death. And we also looked at it in a mouse model of ALS as well. So the short answer to your question is yes, we are keen on translating this. I mean, that's really, as a clinician scientist, that's what you really dream of, is doing something at the bench and seeing it come to patients. So I would very much like to see it get to patients. And there are some efforts in the works to do just that. I certainly don't have I don't yet have a time frame, but that is certainly our hope, that we'll be able to bring that to patients, because thus far it looks safe to use. And so we and colleagues really around the country are keen on bringing that to you.
Excellent. So more on your research, you mentioned you're working on stem cells. How far are we from being able to inject healthy stem cells back into patients? And maybe you can tell us more about different types of stem cells. Yeah,
it's a great point. I mean, all stem cells are not created equal. You've probably heard about what we call mesenchymal stem cells that have been taken from bone marrow and have been put into the cerebrospinal fluid of ALS patients. Those are very different from, and you've probably, have been some fetal stem cells transplanted into spinal cord and brain and so forth. Induced pluripotent stem cells are a different ball of wax. And I would tell you this, I think we are, my belief currently is that iPS cells from ALS patients or from healthy control patients currently are probably best utilized to understand disease mechanisms and to design and screen drugs. We call it like a clinical trial in a dish. If we can make stem cells from lots of patients, which we've done as part of the Answer ALS effort, from 1,000 individuals, can we start to understand what drugs affect different patients just by doing some of those studies in a dish? So my current belief is that's probably the best way to use induced pluripotent stem cells currently for drug discovery, because I think there's really good drugs out there.
Wait, there's a number of questions, very scientific, very detailed. You talked about cryptic peptides. So can you explain again how they affect the beginning of ALS and the progression of ALS?
Yeah, it's a great point. So there are some differences in one particular protein that's affected by TDP-43 called Stathmin-2, and that's really under intense investigation because it seems to be a protein that helps nerve cells send out axons. And so there are some efforts now to try to understand that particular protein. It may be that a lot of the cryptic peptides don't necessarily have anything to do with ALS progression, but they might be just road signs, if you will, and we can use them in the spinal fluid to try to understand when TDP-43 isn't working very well. So it may be, there's a bit of a technical issue there, but it may be that the ones I showed you, for example, aren't particularly related to ALS itself, but they're just flags to say, look, TDP-43 isn't working right because there are other ways or other proteins that TDP-43 might be affecting.
So you spoke about the central role of TDP-43. So the question is, would it be best to keep TDP-43 in the nucleus, or would it be best to sort of try to remove it once it's in the cytoplasm?
That's a great question. There are research efforts going on in both directions. I think the preference, if you will, and that is if it's feasible, would be to keep TDP-43 where it started, and that is in the nucleus. We have, there's evidence to suggest that TDP leaks out or gets mislocalized out of the nucleus, potentially even long before disease even starts. So keeping it in the nucleus where it belongs is probably a way of preventing maybe disease from starting in the first place. There are efforts to say once it gets into the cytoplasm and forms these aggregates, if we dissolve those aggregates or stuff it back into the nucleus, let's say, could that work as well? And there are a number of efforts going on along those lines as well. So I think the answer is potentially both, but I think like to keep it where it started.
Yeah, so one of the attendees made the observation that RNA missplicing is common across different neurodegenerative diseases. So the question is, how specific is this to ALS, and how specific is the myo-18 cryptic peptide for ALS?
Yeah, it's probably, well, I don't know the answer specifically. It was studied in not only ALS, but frontotemporal dementia, so patients with both frontotemporal dementia and ALS. So we don't think it's necessarily specific to ALS. And in fact, we know that TDP-43 abnormalities are also present even in, I think, up to 40% of Alzheimer's disease as well. So it may be that it's not, these cryptic peptides are maybe not specific to any one particular disease, but again, maybe flags for the disease.
One question about a different mechanism. Are you encouraged by PIK5 suppression?
So, I mean, these are really technical questions. I think there are a lot of potential. I think there are a lot of potential candidates, and so I would say that's one of many as far as PIK5 suppression and others. That would be really a short answer, because I think we're just scratching the surface with regard to how how some of these different markers can be suppressed.
Yeah, last question for you, and then I'll ask you some, just to close, just some general comments about the state of the field. But oh, couple of more questions, sorry. And so there's a follow-up question to a previous question about stem cells. So you spoke about induced pluripotent stem cells and how they're best probably reserved right now for testing of drugs. And the specific question is whether other types of stem cells, such as embryonic stem cells, could be used.
I would say that embryonic stem cells or cells derived from embryonic stem cells could potentially be useful. I think since the rise of induced pluripotent stem cell technologies, that those have probably surpassed the use of embryonic stem cells. So I think the future is probably in iPS cell biology, because we can essentially do many similar things to what can be done with embryonic stem cells.
Right. So just last question. I know we always have a lot of attendees who have very specific scientific questions, and that's why we wanted to bring a scientist like you on this webinar, and we'll have your colleague, Dr. Austin, next week. So question about misfolded TDP-43. You mentioned how important it is in the nerves, obviously, as a marker of ALS. So the question is misfolded TDP-43 also accumulated in the nerves of the skin, so that can you actually study the skin and have easy access to that as a biomarker?
Yeah, it's a great point. So I think the answer is potentially. TDP-43 is really it's a ubiquitous protein. It's present in all kinds of different cells. So the answer is yes, that's potentially the case. What it may not tell us is what's going on in the brain. So it's entirely possible that different cells are having different effects. So nerve cells may be very different than skin cells in the way TDP-43 mislocalization occurs, because we don't understand why don't skin cells get sick, for example, and why don't skin cells die? At least not that's very obvious. So it's a great question. TDP-43 is definitely there, but could the signatures be different? And I think that's why CSF has been so attractive.
Great. And now to close, I would like to thank the audience for sending nice messages, thanking Catherine and Jess, you are organizing these webinars and really scheduling our panelists, and thank you to Dr. Maragakis, as one of our attendees says, your time and information gives so much hope, and I would agree with that. And with that, I would like to give you the last word and tell us again, just to summarize the excitement about these new biomarkers and what they could do for ALS studies.
Oh, I would just say we remain enthusiastic. There's a lot of work to be done, but we've come such a long way in really trying to understand ALS. And I think the HEALEY platform is a great opportunity to treat patients, number one, but also to learn from each other with regard to how to do clinical trials, how to do the best research, and how to learn from each other.
Wonderful. Thank you so much. And we'll be back with more information about the trial and also more science next week. Thank you for the great questions as always. Thank you.
Thank you.